in our last meeting? Anyone remember? Nobody. Anyone remember where we stopped last time? Sorry.
At beta chi. At the beginning of beta chi.
The beginning of beta. All right. Okay. Yeah. So we discussed about the alpha structure. Yeah. So alpha structure is the most prominent structure in the protein. So when you have alpha structure in the protein, it's very easy to see. Yeah. It's very easy to see the alpha structure when you have alpha in your protein. Yeah. And the next secondary structure. So alpha, beta turn are secondary structure. Yeah, the primary structure is the sequence of the protein. Yeah, and the alpha, beta, and turn are the secondary. Yeah, secondary structure of the protein. Yeah, so from this sequence, we will arrange into secondary structure. Yeah. So let's look at beta. Yeah. So so it's a zigzag. Yeah, it has a zigzag structure. Yeah. So backbone is extended into a zigzag. Yeah. So there are hydrogen bonds between segments. Yeah, and segments are usually near each other. Yeah. And R groups of adjacent amino acid come out opposite direction. Yeah. And for example, it's a keratin, it's your hair, it's a keratin, a spider web, a keratin. So normally they have a high content of glycine and alanine. Yeah. So it's a small. These are amino acid with small side chain group. Yeah. When you look at glycine, yeah, the side chain is only one hydrogen atom, and for alanine, the side chain is only one methyl group. Yeah. So it's a pretty small amino acid. So normally for beta structure, they have a small amino acid. Yeah. Okay. So we have two beta sheet. Yeah, either parallel or anti-parallel. Yeah. In parallel beta sheets, the hydrogen bond strands run in the same direction. Yeah. So basically, it's resulting in a weaker hydrogen bond. Yeah, that's for parallel. Yeah. For anti-parallel, the beta sheets, the hydrogen bond strands run in opposite direction. So it results in a stronger hydrogen bond. Yeah. So let's look at anti-parallel. Yeah. This is the anti-parallel beta sheet. So basically, it's stronger. Yeah, the hydrogen bond is stronger. As you can see there, yeah, that's a hydrogen bond between CO. Yeah, yeah, between CO. Yeah, and the NH. Yeah. So the blue one is a nitrogen. So that's the convention. Yeah, that's the convention. When you write when you draw the amino acid, oxygen is red. Yeah, nitrogen is blue. Yeah, hydrogen is white. Yeah, carbon sometimes people put gray, sometimes people put green. You know, but so people use not only one color, but oxygen for sure is red. Yeah, for nitrogen is blue. It's always blue, and for hydrogen is always white. Only for carbon people put different colors. Okay. Now, as you can see there, yeah. As you can see there, you can see a hydrogen bond between NH, yeah, and the CO, yeah. So that's for anti-parallel. But for parallel, the hydrogen bond is weaker, yeah, because as you can see, it creates an angle. Once the hydrogen bond creates an angle, then the hydrogen bond becomes weaker. Yeah, becomes weak. So when you compare anti-parallel and parallel, actually the anti-parallel has a stronger hydrogen bond. Okay, compared to parallel beta sheet. Yeah. As you can see here as well, the direction of beta sheet also is different. Yeah, in parallel beta sheet, they go to the same direction. Yeah, to the right, as you can see there, the direction of the arrow. Yeah, all go to the right. But in anti-parallel, they go to opposite direction. Yeah, so that's the difference. Okay, but in terms of their function, they are the same. Okay, right. Now, so after alpha and beta, yeah, there is turn structure. Yeah, there is turn structure, and we call this structure, yeah, as alpha, beta, and gamma. There are different turns, yeah, alpha, beta, and gamma, depending on the size of the turn. Yeah, so the most common is beta turn. Yeah, that's the most common. Yeah, beta turn. Yeah. So it consists of three amino acids, if I'm not mistaken. Yeah. So beta turns occur frequently whenever strands in beta sheet change the direction. Yeah. So when you have beta goes to one direction, all right, and then it turns to create another beta. So that turn is called beta turn. Yeah. Because you saw before, yeah, beta sheet consists of different sheets. Yeah. So whenever they change the direction, yeah, to create another beta sheet, they are connected through turn. Yeah. So we call this turn as beta turn. Yeah. And also it connects different alpha helix as well. Yeah. So it connects beta between beta sheets. Yeah. So when you have one beta, you have the second beta is connected through turns. So the same thing with alpha. When you have the first alpha and then the second alpha helix, it's connected. They are connected through turns. Yeah. So 180 turns. Yeah. So the turn is 180. Yeah. It's accomplished over 4 amino acids. Yeah. The smallest is 3, but 3 is quite rare. Yeah. So 4 is more common. Yeah. 4 amino acids. 1, 2, 3, 4. They create turn. Yeah. And then they start with another beta sheet or another alpha helix. Right. So 2 alpha helix or 2 beta sheets are connected through beta turn. And the size of this beta turn is between 3 or 4 amino acids, but 4 amino acids are more common. Yeah. So the turn is stabilized by hydrogen bond between amino acid number 1 and amino acid number 4. Yeah. And the position number 2, mostly they have proline. Yeah. Because you saw proline before. Proline can undergo a transformation from cis to trans. Yeah, cis trans. I don't know whether I saw it to you. Let's see. Yeah, I think in the next slide. Yeah. So in position number 2 or 3, there is proline or glycine. Yeah. The reason why is because proline and glycine are very flexible. The flexibility of proline comes from cis and trans isomers. Yeah, I'll show you in the next slide. Yeah. And glycine, the flexibility of glycine comes from the size of the side chain. Glycine is the smallest amino acid. Among 20 amino acids, glycine is the smallest. Yeah, with only one atom for the side chain. Yeah, only one atom, which is hydrogen. So that's why it's flexible. So this flexibility helps the formation of beta turn because in beta turn they have to turn. Yeah, they can belok. Yeah, they have to turn. Yeah. So they need flexible amino acids, which are proline and glycine. All right. So this is turn. Yeah. Look at the type 1 beta turn. Yeah. 1, 2, 3, 4. 4 amino acids. Yeah. 1, 2, 3, 4. It creates a turn, right? You can see the turn. Yeah. 1, 2, 3, 4. That's proline at position number 2. Yeah. 1, 2, 3, 4. And there is a hydrogen bond, yeah, in the middle to stabilize the turn. So the turn will not open. Yeah. It stabilizes the turn. And proline at position number 2, because as I mentioned before, proline is very flexible. Okay. And this is type 2 beta turn, 1, 2, 3, 4, 4 amino acid as well, with hydrogen bond in the middle and glycine at position number 3. And as I mentioned before, glycine is the smallest amino acid. That's why it's very flexible with only one. Yeah, with only one atom at the side chain, which is hydrogen. You can see the white dot here. Yeah. So the white one, I need to put pen. That one. Yeah. That's the side chain of glycine. All right. Smallest amino acid. That's why it's flexible. But for proline, yeah, it has this ring. This ring is very flexible. You can see my drawing, right? I draw it red. You can see my drawing, right?
Yeah.
Okay, all right. So that's the difference between proline and glycine. Okay, all right. So most prolines, yeah, they have trans configuration, yeah, because that's the most stable one, yeah. But when you have cis configuration, yeah, there are cis configuration as well. That's the minority. It's only 6%. Yeah. So most of the 6% involve beta turn. Yeah. But in most cases, when proline is not located in turn structure, they will have trans configuration. Yeah. But when proline is involved in the formation of beta turn, then proline will change into cis configuration. Okay. Yeah. So you can see here, yeah, this is trans. Yeah. This is trans. All right, and this is cis. Yeah, you can see the difference. Yeah, you can see the difference. Yeah, follow this bond. Yeah, this is trans. Okay, and this is cis. Which one is the most stable? Of course, trans is more stable compared to cis. Yeah, but the changes between trans and cis sometimes happen spontaneously. Yeah, sometimes it happens spontaneously. Yeah. So when you have a peptide or when you have a protein and you have proline, all right, you may expect that cis trans changes could happen spontaneously. Yeah, it could happen spontaneously, even though cis configuration is less common compared to trans. Okay, so that's the reason why when you have proline in alpha helix, it will break your alpha helix. Yeah, because of this change from cis to trans or from trans to cis. Yeah. It happens spontaneously. You see this signal, you see this symbol. Yeah, it's equilibrium. So it happens spontaneously. All right. So another term is gamma turn. It's less common. Yeah, 3 amino acid or less. Yeah, it's smaller turn. Yeah, it involves only 3 residues. Yeah. So it consists of gamma turn and inverse turn. Yeah. So there is a hydrogen bond between amino acid number 1 and amino acid number 2. Amino acid number 1 and amino acid number 3. Okay. So there is a hydrogen bond. So it's less common. Yeah. Because it's small. When you have small turn, it requires larger energy to create the turn. Yeah, because it's quite difficult to form 3 amino acid into one cyclic structure. It's not easy. Yeah? Just like when you drive. Yeah, when you drive, when you have to make small turn, it's quite difficult, right? I don't know whether you can drive. Yeah? I don't know whether your age is you eligible to drive to have a driving license. I don't know. But when you make small turn, yeah, for example, in the traffic light, yeah, you have to make very small turn, yeah, because you want to go to another direction. You have to make very small turn. Of course, it's more difficult, right? But when you drive in a roundabout, yeah, very big turn, it's easier, right? Yeah. So the same thing for gamma turn is less common because it involves only 3 amino acids. and the energy required to create these three amino acid cyclic structure is higher than beta 10. Yeah. So beta 10 is more common. Okay. Right. Okay. So this is one way to analyze a protein structure. Yeah. Using a circular dichroism. Yeah, to see the secondary structure of the protein. Yeah. We will discuss in detail in our next lecture, yeah, when we discuss spectroscopic method in protein analysis. Yeah. But for this one, I just discuss briefly. Yeah. So we use this technique to see the secondary structure of a protein. Yeah, your protein. Yeah. So it measures alpha, beta, and tern. Yeah. Because in your protein you have chromophores. Yeah, you have chromophore, and then these chromophores can absorb UV light. Okay, in the chiral environment, yeah, it has a chirality. Yeah, it has chirality because amino acids are chiral molecules, so it can absorb UV signal in a very unique way, so that you can differentiate between alpha, beta, and theta. Yeah. So this is the typical, yeah, spectrum of CD, yeah, cyclodichroism. Yeah. When your protein has alpha helix, you will see the absorption. You will see the absorption. Yeah. You will see the spectrum like the blue line. Yeah. That's alpha helix. A very typical alpha helix. Very easy to see. Yeah. When you have beta, when your protein has beta conformation, then you will see the red line. All right. But if your protein is random, just random. Yeah. It's not alpha, it's not beta. So you will see the black line. The problem is, in most cases, the protein is mixed. It has a little bit of alpha, it has a little bit of beta, and it has a little bit of random. Because as I mentioned before, when you have alpha or helix, when you have alpha or beta, you have several alpha and several beta connected through turn, right? They are connected through turn, and this turn structure is considered as random. Yeah, the turn structure that connects two alpha helices or it connects two beta shapes. Yeah, either a gamma turn or beta turn, they are random. So in most cases, you will have a mixture of alpha, beta, and random coil. If that happens, cyclodichroism can differentiate them. Can. That's a good thing about cyclodichroism. That's the reason why 15 years ago we decided to buy cyclodichroism, but not in chemistry department. It's in biotechnology, yeah, in faculty of biotech, because we bought it last time. Yeah, I was the first person in Malaysia who did research using cyclodichroism, because I did protein research 20 years ago. And then I still remember at that time, we want to see the secondary structure of the protein, but we don't have the equipment. So we decided to buy, and we have the first cyclodichroism spectrophotometer, yeah, in Malaysia. We bought it 15 years ago. Still running very well until now. It's very easy to use, yeah, but difficult to maintain. Yeah. Easy to use, but difficult to maintain. But by using CD, you can see the secondary structure of the protein, yeah, and it will give you a ratio. How many percent alpha, how many percent beta, how many percent random coil. Yeah, it will give you a very precise information. How many percent alpha, how many percent beta, how many percent random coil. And normally we use CD when we want to see the changes in protein structure. For example, we heat the protein. We put the protein in high temperature. And as we discussed last time, when you heat the protein, you lose the structure, right? You lose the structure, you lose the alpha, you lose the beta, you get more random coil. you lose alpha, you lose beta, you get more and more. So we can follow that using CD, because in CD they have heating plate. You can heat the sample in CD. Even you can cool down the sample in CD. So it's equipped with heating plate, and also it's equipped with a cooling plate. Yeah, so it's very sophisticated machine. I like it so much. So you can change the temperature. When you change the temperature, you will see some changes. CD can detect the changes. Yeah. So when you heat the protein starts losing the alpha structure. It starts losing the beta structure, but it starts getting more random structure. So you can predict the stability of the protein. We did quite a lot of research on this, yeah, because we work on thermostable protein. Thermostable protein, the protein that we obtain from hot spring. We have hot spring in Malaysia, yeah. So the protein that is stable to high temperature. This is a very important protein in industry, because in industry they use high temperature. So they need enzyme that can work in high temperature. For example, in the sugar industry, yeah, because the kind of sugar that they use is fructose, and fructose is very thick, liquid, but it's thick. Yeah, so they have to flow the fructose inside the pipe, and to make sure that the fructose doesn't stuck inside the pipe, they have to increase the temperature. So the sugar, yeah, the very thick sugar can flow inside the pipe, right? So they use high temperature, yeah, and also they use enzyme as well for enzymatic reaction, yeah, to change sucrose into fructose. Yeah, they need enzyme, and enzyme is protein. And we know that protein is not stable in high temperature. So that's why we try to look for enzymes from hot spring, from microbes in hot spring, yeah, or from volcanoes. Yeah, if volcanoes, maybe we don't have volcano in Malaysia, we have to go to other countries. We isolate the protein from there, and we check the stability. We increase the temperature until 50, 60, 70, and we see whether or not the protein loses the structure. If the protein doesn't lose the structure at high temperature, we call this protein as thermostable protein. Yeah, thermostable protein. And how we monitor it? Using CD. If in high temperature the protein starts losing the structure, then the protein is not thermostable. And it's amazing to see that some protein can survive in high temperature. Yeah, because all this protein, we isolate this protein from hot spring, from volcanoes, So basically, organisms can survive in high temperature. Yeah. Because what? Because the protein that they have in their cells can stand high temperature, can survive in high temperature. Not like us. We don't have thermostable protein in our body. At 40 degrees Celsius, the protein starts losing the structure. Yeah, but those microbes, bacteria, viruses in hot spring, in volcanoes, there are lives. When you go to volcano, when you go to hot spring, there are organisms. You isolate the organism, you isolate the protein from their cells. You check using CD to increase the temperature. At 60, 70 degrees Celsius, they maintain the structure. They don't lose their alpha, they don't lose their beta. Amazing. Not only that, we did a lot of research on protein. I remember last time we sent a team to Antarctica, South Pole. Yeah, we isolate some organisms from there, bacteria, yeast, some microbes. Yeah, and we isolate the protein from the cell to understand why they can survive in low temperature. Yeah, we call this organism as cyclophilic. Yeah, cyclo is low temperature, philic is love. So the organism can survive in low temperature. And we discovered several proteins that help this organism to survive in low temperature. We call this as antifreeze protein. And this is the research that first I did in UPM 20 years ago, antifreeze protein or AFP. We published several papers on AFP, the protein that was isolated from organisms from South Pole. Yeah. UPM scientists died during the mission. Chief Omar from the Department of Biochem when he was diving. Yeah, and he was diving for some reason. There was accident and he died during the mission. Yeah. So it is quite sad, basically. Yeah. The first and the only Malaysian died during the mission in South Pole. But we brought back the organisms. Yeah. We are able to isolate the proteins, and we know why these organisms can survive at low temperature, because their blood is not frozen in low temperature. Their cells is not frozen in low temperature, because they have this very unique protein called antifreeze protein or AFP that maintain their blood in liquid form, that maintain the integrity of cells even though in low temperature. And we are able to know the structure, we are able to study it, and I published several papers on that. Now we use the protein for preservation, yeah, to preserve the cells, to preserve the organ in low temperature. We use this protein. Yeah, we work together with professors from faculty of veterinary. Yeah, and we have published several papers as well. Yeah, so a lot of things you can do with CD. Yeah, and yeah, I'm sorry I took your time several minutes just to talk about CD, because I'm very passionate about this instrument. Yeah, I use this instrument a lot for my research. Okay, so any questions? No questions. All right, so we are done with secondary structure. We are done with primary structure, secondary structure, now tertiary structure. Yeah. So basically, tertiary structure is the overall spatial arrangement of atom in a protein. So it's an arrangement of different secondary structures in a protein. So basically, the whole protein. Yeah. Secondary structure is just one part of the protein, either alpha or beta. When you combine them, it becomes a whole protein. Yeah. So all the structure that you see, yeah, from one protein is tertiary structure. Yeah. So this structure is stabilized by numerous weak interactions. Yeah. Mostly hydrophobic and polar interaction, and some by disulfide bond as well. Interacting amino acids are not necessarily next to each other, yeah, in the primary structure, not necessarily. Sometimes they are far, but because of their spatial arrangement, yeah, they are close to each other, and they create some interaction to stabilize the tertiary structure. And they can be classified into two major classes. One is fibrous protein, and the second one is globular protein. All right. So fibrous protein is water insoluble. Yeah, it's hydrophobic, and the best example is your hair. It's fibrous protein. You need this for structure and shape of cells, for the integrity of cells. Yeah, because fibrous protein is very strong. Yeah. Keratin, your skin. Yeah, your skin consists of a lot of keratin. Keratin is protein in your hair. It's protein as well. Yeah. And the second one is globular protein. Yeah. Enzymes are protein. All the proteins that you have that regulate your metabolism, they're globular protein. They're water soluble. They are water soluble. All right. So the proteins that you see on the surface of your skin, on hair, all the fibrous protein, yeah, they are very strong, very strong, yeah? Because we need them to maintain the integrity of cells. Okay, for fibrous protein, yeah, it's tough, yeah, it's rigid, yeah. This cross-linked alpha helix, you have keratin, yeah? your nails, horns, collagen on your skin, that's protein. Yeah? So that's why if you want to maintain your skin, you don't want to get wrinkle, you need to consume a lot of protein with collagen. Yeah? Silk, fibroid, spider web. It's soft, flexible, and then exact nest web. So that's those are examples of fibrous protein. Right. So fibrous protein, yeah, it has properties that give strength and flexibility. Again, all fibrous protein are insoluble because they have plenty of hydrophobic amino acids inside and on protein surface. Alpha keratin is the most common example of fibrous protein. Yeah. This is the structure of alpha keratin in your hair. Yeah, you can start seeing your hair now. Take a mirror and see your hair. So when you zoom in, this is the structure. It's beautiful structure. Yeah. So it's a coils assembled into large supramolecular complexes. Yeah. And when you zoom in again, the structure is like this. Yeah. So this is your hair. Right? It has an alpha helix structure. Yeah. And it's angled to create a supramolecular structure. Right? So that's your hair. And sometimes you want to change your hairstyle. So basically, this is what happens. Yeah. So your hair, the integrity of your hair is maintained by this bond called disulfide bond between two S. Yeah, two sulfur. So we call this as disulfide bond. So the first thing that in hair salon, the first thing they do to your hair is to reduce this, to break this disulfide bond into SH. Right? They break it. Okay? And then they change the shape of your hair. Alright? By doing something like that. Yeah? They change the shape, And then once you get the curliness that you want, the shape that you want, and then the disulfide bond is reoxidized. You will get a new disulfide bond, but now it's different. Yeah? The shape of your hair is different now because it's different from the disulfide hair that happened initially. Right? So there is a rearrangement of disulfide bond. So this is what happens when you change your hairstyle. Yeah, you make it curly. Initially you have straight, and then you make it curly. This is what happens. So that's the reason why when you walk through hair salon, you smell something unpleasant. Yeah, something very unpleasant, right? Bau busuk macam I don't like the smell. When you realize, yeah, whenever you walk through hair salon, you can smell that. That's the chemical they use to reduce the disulfide bond. We call it as a reducing agent. And reducing agent, the smell is pretty bad. I knew it because I work in the lab a lot using reducing agent. And the smell is horrible. Yeah, I don't like the smell. Yeah. So whenever I walk to a hair salon, I can smell that familiar, aroma. I know, well, they use this chemical to reduce the disulfide bond into SH. And then they reoxidize to create a new disulfide bond. Okay. Any questions about your hair?
If people have curly hair,
Yes.
how?
If you have curly hair, you can straighten it. you can straighten your hair by changing this. Yeah. Okay. Put the reducing agent and then make it straight and then re-oxidize. Can. Why? You're interested? Can. Either way it can be done. Yeah, either way, from straight to curly, from curly to straight. It's just about rearranging the sulfide bond. The rearrangement of the sulfide bond. Okay. So when straight to make it curly, first you reduce the sulfide bond. Yeah. And then you shape the hair, put the oxidizing agent, then you got new hairstyle. If your hair is naturally curly, you want to make it straight, put reducing agent, and then straighten your hair, put oxidizing agent. That's it. So either way it can be done. Alright? Botak pun boleh potong, but you don't need reducing agent. Just cut. So yeah, that's the magic of chemistry. Right? Any other questions about your hair? No, right. So keratin is the most common protein that you can find in your environment. Yeah, all those dust that you see on the surface of your table, that's the keratin. Even though you close the room, you don't allow dust from outside to enter your room. But after 1 or 2 days, you realize that there are a lot of dust right on your table. It comes from your skin because your skin has to regenerate, right? By removing the dead skin. Yeah. So it goes to the air, and then it will fall down to your table. So when you see dust on your table, mostly it's keratin from your dead skin. Yeah, it's keratin. You can analyze it. Even I did some analysis accidentally, in 2007. Yeah, so it's 13 years ago when I worked on this project in the US. So I worked on this protein, and I remember during that time, during winter, so it's very cold, and during winter our skin is very dry. Yeah, our skin is very dry, our hair is very dry, and I don't even cover my hair, and some of my skin is exposed. So when I work in the lab, the keratin from my skin falls to my sample. Yeah, accidentally. And I analyze my sample using mass spectrometry, very sensitive way to analyze the protein, and I see a lot of keratin in my sample. Initially, I don't realize that, because I'm doing research on cancer cells, and then I report to my boss, "Hey boss, my cancer cells has a lot of keratin, My boss just laughed at me. Meanwhile, this is winter. You contaminate your sample, it's contaminated from your skin, from your hair. Cancer cells doesn't have keratin, it's from your skin, from your hair. So well, because that's the most common protein that we can find everywhere. It comes from your body. It comes from your dead skin. Okay. All right. Next is collagen. Collagen is if you have collagen in connective tissue, in tendons, cartilage, bones, cornea of the eye, So normally it has long glycine and proline, and it's left-handed helix. The triple helix has higher tensile strength than the steel wire of equal cross-section, so basically it's very strong. So typical collagen has around 35% glycine, 11% alanine, and 21% proline. So the amino acid sequence is generally a tripeptide, glycine XX. So there is an enzyme that can break down collagen. It's called collagenase. So this is collagen. Yeah, this is collagen. Okay. So the structure is a helix. Yeah. From the top you can see. So it's very, very beautiful. And this is when you see the collagen through microscope. Okay. Okay, what about silk? Yeah. Silk is a fibrous protein as well. It has anti-parallel beta structure, so that's why it's very strong. They have small amino acids, alanine and glycine, so it allows for close packing of sheets. Yeah, so that's why the packing is very close because the amino acid is small, alanine and glycine. It doesn't stretch too much, yeah, even though it's flexible. So the structure is stabilized by a lot of hydrogen bonds, yeah, and also London dispersion. So it's very weak interaction, but because the packing is very nice, so the result is a very, very strong structure. Yeah. Yeah, this is the structure of a silk, of a spine cap, silk. It's anti-parallel beta structure. All right. So it's 10:50 now. Let's have a break for 5 minutes maybe, and then we will continue. We go to the protein. Okay.